Hey guys, welcome back to Unstop Biology. So guys, in today's video, we are going to cover where exactly the ATP and NADPH, which is produced during cyclic or non-cyclic photophosphorylation, which is produced during the electron transport system, is consumed. What is the usage of these ATP and NADPH, right? So till now, what we have studied is that these these products of light reaction are what? It is either ATP or NADPH or O2, right? So we have ATP or NADPH or oxygen molecule, which is released as products of light reaction. Now, out of these, this oxygen, this diffuses out of chloroplast, right? So this basically goes out of chloroplast, right? While the ATP and NADPH, both of these are used to drive the processes which leads to synthesis of food, which leads to synthesis of food, right? So what are these food items? These are basically sugars. And this process is known as biosynthetic phase, right? So this phase is known as biosynthetic phase of photosynthesis, right? This phase in particularly is known as biosynthetic phase of photosynthesis. Now, this process basically does not directly depend upon the presence of light, but is dependent on the products of the light reaction, which is ATP and NADPH, right? So, apart from CO2 and H2O, these energy molecules are required to convert CO2 and H2O into sugar, right? Now let us see how the ATP and NADPH are exactly used in the biosynthetic system. Now we earlier saw that carbon dioxide is combined with water molecule to form sugar, right? Now basically there are several cycles, guys. There are several cycles which are involved in this conversion of the inorganic carbon dioxide or water molecule into sugar, right? So basically one of the biosynthetic pathway, these cycles are known as various biosynthetic pathways. So one of the biosynthetic pathway is Kelvin cycle, right? Which is where the first product is 3-phosphoglyceric acid, which is in short known as PGA, and this cycle is also known as C3 pathway, right? So the plants with first product as C3 acid follows the process of C3 pathway. You'll have to remember that, right? The other cycle involves basically formation of the first product as 4 carbon product, and we'll study that particular cycle in the next video, right? Now what is the primary acceptor of carbon dioxide? Here we'll have to understand that as well, right? So basically if you see here, the acceptor molecule, the carbon dioxide acceptor molecule was a 5 carbon ketose sugar. The molecule is a 5 carbon ketose sugar, and it is known as ribulose bisphosphate. In short, RUBP. Right? So this is the particular acceptor molecule of carbon dioxide. Now let's see what exactly is the Kelvin cycle. So it is named after a scientist, guys, right? It is named after a scientist, and the scientist Melvin Kelvin. So Melvin Kelvin, along with his co-workers, along with his partners, worked on the entire pathway and showed that this pathway operated in a cyclic manner, right? This is a cyclic pathway, and here ribulose bisphosphate was generated, right? Now let us understand how this pathway operates and exactly where the sugar is synthesized, right? Now, first you'll have to understand very clearly that Calvin pathway occurs in all photosynthetic plants. It occurs in all photosynthetic plants. So it does not matter whether they have C3 or the C4 pathway, but they should be a photosynthetic plant, right? So, in order to understand the entire Calvin cycle, we basically provide three different stages of the cycle. Now, the first stage of Calvin cycle is known as carboxylation. The second stage is known as reduction. And the third stage is known as regeneration. Right? So now let's do one thing. Let's understand this entire process through this particular cycle itself. Let's see one by one what happens in the three stages. Okay, so this is the cycle, guys. So I hope it is clearly visible to you. Now, first of all, what is happening is carboxylation. So the first process is of carboxylation. Now here, there is a fixation of CO2 into a stable organic intermediate. So first, in the first step, as it itself is called as carboxylation, here we are fixing the carbon dioxide, right, into a stable organic. It is an unstable inorganic compound. We are now converting it to a stable organic intermediate. So basically carboxylation is known as the most crucial step of the Calvin cycle, where we utilize carbon dioxide for carboxylation of ribulose 1,5 bisphosphate. So we are using RUBP, right? We are using RUBP, right? So for carboxylation of RUBP, we are basically using the carbon dioxide molecule. And this reaction is catalyzed by an enzyme which is known as RUBP carboxylase. So it is also being catalyzed by the enzyme RUBP carboxylase, which now this results. So basically, we have atmosphere. From atmosphere, we are getting the carbon dioxide and water molecule. Now here we are fixing this carbon dioxide by carboxylation of RUBP in the presence of the enzyme RUBP carboxylase, and this results in the formation of two molecules. Remember, two molecules are formed of phosphoglycerate, which is known as PGA, which is also known as phosphoglyceric acid. So now from ribulose 1,5 bisphosphate, the first compound that we get, the first thing that we get is 3-phosphoglyceric acid or 3-phosphoglycerate. Now, since the enzyme, the Rubisco, the RUBP carboxylase enzyme, has also an oxygenation activity, right? It would be more correct to call it as RUBP carboxylase oxygenase, and that is why it is known as Rubisco, which is RUBP carboxylase oxygenase, right? Now the first step is clear. Now from here starts the second step, which is reduction. The second step is reduction. So now here again there are a series of reactions that result in the formation of glucose. So basically this steps involves utilization of also two molecules of ATP. Right? So here we have ATP and NADPH utilized, which gets converted to ADP and NADP plus phosphate, inorganic phosphate, right? So here we are using energy. Now here what happens that basically the two molecules of ATP and two molecules of NADPH are basically getting used for reduction per carbon dioxide molecule. So basically fixation of six molecules of carbon dioxide and six turns of the cycle. Okay, total six molecules of carbon dioxide and six turns of this cycle are required for formation of one molecule of sucrose, basically glucose first, right? So for one molecule of glucose, you require six, right? You require six molecule of carbon dioxide and six cycle of this. Right? So basically here what is happening, your phosphoglyceric acid or 3-phosphoglycerate is getting converted into triose phosphate, and we release the molecule of sugar as sucrose or as starch. Now, this starch gets either stored or utilized. Now, what happens to the triose phosphate? So, now basically we have to regenerate, right? We have to, since it is a cyclic process, we have to regenerate the ribulose 1,5 bisphosphate back from triose phosphate, right? So here basically regeneration of the CO2 acceptor molecule, which is RUBP, is crucial so that the cycle is not interrupted, right? So the regeneration steps require again one ATP. So again one ATP is converted to ADP, and this is the third stage, which is of regeneration, right? And again here phosphorylation happens, right? So ATP is required for phosphorylation, and we get RUBP, ribulose bisphosphate, from triose phosphate. Okay, so again basically if we talk about the total process, what we are right now monitoring. So every CO2 molecule enters the Calvin cycle, then we utilize three, two here and one here. So we are utilizing three molecules of ATP and two molecules of NADPH, right? And this basically meets this difference in number of ATP and NADPH, which is used in the dark reaction. This is how they are produced in the dark reaction as well, that the cyclic phosphorylation takes place, right? So basically, sixth molecule, if I talk about what goes in and what comes out, basically in terms of ADP and the product, what is happening here? Here, basically six molecules, right? Six molecules of CO2 is going in, 18 ATP, because six cycle are happening, and in every cycle we are using 3 ATP. So six cycles, 3 6 are 18, and then two molecules of NADPH and then again six cycle. So 12 molecule of NADPH. This goes in, and then we get one glucose ka molecule and 18 ADP and 12 NADP. So this is what comes out. And guys, this is the entire process where carbon dioxide and water is utilized and is converted to a sugar. So I hope you would have understood this entire process of Calvin cycle. If not, guys, please do not hesitate. I would request again to not hesitate in asking questions. You can ask all your doubts either by just mentioning in in the comment of this video, or you can also ask your doubt through WhatsApp. I have given the WhatsApp number in description. And guys, if you did understand the video, please do like it and share it with your friends. And if you are new to the channel, please do subscribe to the channel and press the bell icon to get all the updates that I'll be posting on this channel. Thanks for watching this video, guys. Take care till then. Bye bye.